All right, so the first thing we're going to talk about, or the only thing we're going to talk about in this video, is what these scientists did. And so the first scientist we're talking about is Frederick Griffith, and he was working in the early 1900s, and he discovered the process of transformation, or he named that process. And so what he did, what transformation is where one bacteria, in his case, what he was talking about, one bacteria gives some information to another bacteria, and transforms it. So it changes. Think about what it means to transform. Think about like the Transformers. What they do, they transform shape and therefore function. And so the same thing here. By giving that new information, they transform the structure of the organism, in this case bacteria, and therefore the function. So what he did is he gave mice this smooth-coated bacteria, smooth-coated because it had an extra capsule thing around it, and the mouse died. Then he gave them the rough-coated bacteria because it was missing that capsule, so it was not smooth. And the mouse was able to be healthy because the immune system could see that bacteria and could kill it. Then he killed the S cells, the smooth ones that had the capsule around it, killed those with heat, injected it into the mouse. No big deal because it was dead. Not the mouse, this bacteria was dead. And so then he mixed the two together. The heat killed smooth cells, which were not a problem because they were dead, and living rough cells, which were not a problem because the mouse's immune system could see them. But somehow there was a transfer of information, and now the rough cells started making that smooth capsule and were able to evade detection by the immune system, and so they killed the mouse. And so when he tested the mouse, what he found is that those R cells had been converted to S cells. They had been given that capsule and therefore were smooth. And so he again discovered that something was being transferred from one organism to another, from one bacteria to another, that transformed that bacteria, which caused it to change shape and therefore function. So he's kind of the father of transformation. I don't know if that's a thing, but he's the one that coined that term. All right, then a couple of years later, about 15 years later, Avery, McCarty, and MacLeod decided to, and I'm going to fix that because that's going to bother me, there we go, decided to try to figure out what was the transforming factor, because they knew that DNA was important, they knew that RNA was important, they knew that protein was important, but they didn't know what did what at this point. So they went in and they inactivated DNA, and when they did that, the transformation didn't occur. They said, okay, well, that makes us think it's DNA, but we still need to check RNA and protein. Maybe it's more than one of them. And so then they repeated their experiment, and this time they inactivated the RNA. Well, transformation still happened. So they said, well, it must not be the RNA doing transformation because it's still happening and the RNA is deactivated. And then they repeated it a third time, deactivating proteins. And again, transformation still occurred. And so that helped them conclude that DNA and DNA alone was the transforming agent. That was what was making those bacteria change. And so they are credited, Griffith said transformation's a thing. Avery, McCarty, and MacLeod said, yeah, it is a thing, and DNA is responsible for it. So that's kind of their contribution. The next are Hershey and Chase. One of these is Martha. I don't remember. I think Hershey, but I'm not going to promise that. So we're starting to see some females in science, which is always a good thing. And they worked with bacteriophages. Bacteriophages are these adorable little viruses that infect bacteria. And what they wanted to know is they knew that the viruses, the bacteriophages, were made of DNA and protein. They knew that they were basically this protein coat surrounding this core of DNA. And what they wanted to know, though, is which of those was going into the bacteria and infecting it. So what was the genetic material that was being passed? Well, they didn't call it genetic material, but what was the material that was being passed from the virus to the bacteria that was killing the bacteria and turning it into a factory for the viruses? And for us now, we're like, well, duh, it was the DNA. But back then, they didn't know. Like proteins, honestly, proteins were more important than DNA for a long time. They thought proteins did everything until they realized that DNA made proteins. I mean, we still know that proteins do everything, but now we know that DNA makes proteins. So they really didn't think DNA was very important, and they assumed it was proteins back in these days. Like Oswald, Avery, and them, they probably assumed it was protein as well, and nope, it was DNA. So anyway, what they did is they labeled the proteins with radioactive sulfur, because there's no sulfur in DNA, so there's no way that it would accidentally get in there. And they labeled the DNA with radioactive phosphorus, because very rarely do we see phosphorus in proteins. And so, but we see it all the time in DNA in that phosphate backbone, sugar phosphate backbone. And so then they infected the bacteria with the viruses that were labeled, and then they tested the bacteria. And so when they did this, they found that what went into the bacteria was the phosphorus, the radioactive phosphorus, which told them that it was the DNA that was entering the bacteria. So they were able to determine that DNA is the genetic material that is transferred when a virus infects something, and not protein. So again, this is kind of slowly breaking down protein's importance. And now that we know more, we realize that proteins are super important. They do run the world. But DNA is important as well because it codes for those proteins. All right, and then the next one is Chargaff, good old Irwin. And he was just studying some things, some genetics, DNA composition. Now, at this point, they don't know what DNA looks like, okay? But they do know that there's some different parts to it. And so, but they just don't know how those parts are organized, how they're structured. And so what he studied is he started looking at what composition, what percent of adenine am I seeing, and what percent of thymine and guanine and cytosine am I seeing. And when he started studying this, just out of curiosity, that was just his field of study, he realized that every time he studied adenine, he was getting roughly the same numbers for thymine. And when he studied guanine, he was getting roughly the same numbers for cytosine. And he thought, I wonder why that is. And so he came to the conclusion that adenine and thymine bind together. So everywhere there's an adenine, there should also be a thymine, and that's why their numbers were equal. And guanine and cytosine bound together. And so everywhere there's a guanine, there should be a cytosine, and that's why their numbers were equal. And so that's why we call these Chargaff's rules. You'll see that sometimes. You'll see it called the base pairing rules. But it just means that A and T bind together and G and C bind together. All right, and then the last one we're talking about are Franklin, Wilkins, Watson, and Crick. And there's a little bit of tension when you talk about these, especially out in the scientific community, because Rosalind Franklin did a lot of work and she didn't get any credit for it, really. I mean, she is now, like now that people realize what happened, she's getting a lot of credit. But there's a couple of reasons, one of which is she was a female in the 50s. And unfortunately, that's part of what it was. But another reason is that she died before the Nobel Prize was awarded, and you can't get the Nobel Prize if you are deceased. And so if she had still been alive, I don't know what would have happened, because I believe only 3 people can get the Nobel Prize, and there were 4 people that worked on this. So I don't know what would have happened. But anyway, so she worked with Maurice Wilkins, and they took X-ray crystallographs of DNA. And so this is kind of what they saw, looking down from the top of DNA down, and you can see lots of even spacing, so that you don't see anything in here or here or here or here. You don't see a lot in between these spaces. And what this told us is that there was the measurements in the DNA were evenly spaced. And so this was crucial to Watson and Crick determining that it was a double helix structure. I don't know that she had figured out that it was a double helix, but I do know that they read her notes, they read her research that she had published and that she had shared, and that was key to them figuring out the double helix. So I don't know that she said double helix. I don't know of any reference to that, but I know that without her work, they would not have figured that out. So she and Wilkins, I say her work, she gets the most credit for it now, but she and Wilkins were definitely crucial to Watson and Crick figuring that out. She did die of cancer, they think probably from the x-rays. And then we have Watson and Crick. So James Watson and Francis Crick in the early 1950s were the ones that actually came up with the structure, built this 3D model of what DNA looked like. That double helix, that twisted ladder structure with the sugar phosphates on the sides and the bases in the middle. And they, of course, piggybacked off of Franklin and Wilkins X-ray data as well as Chargaff's rules. They knew all of that. So they had all of that information when they were building this. And so they are credited with that. They did give Wilkins the Nobel Prize as well, because his and Franklin's research was crucial to these guys understanding what they figured out. So again, lots of scientists do lots of work, and that's kind of what happens when you have big discoveries. It's usually not just one scientist. Sometimes it is, but usually it's a lot of scientists over a lot of years collecting data and sharing that data and coming to those conclusions. So that is each of the groups of scientists or individual scientists and how they contributed. I hope that helps, but if you have questions, let me know.